Good morning, everyone, and welcome to our foundation and our fall webinar series. This is a recorded session, so I will record this and post this to all of you who want to take a look at this later on. But a big welcome. My name is Cecilia Dahlström, and I am the Secretary General at PAR Foundation. Gabriel Oke is with me today. He's our communication manager, and he will monitor questions and make sure that your questions are seen. We have a tough morning today because maybe it's the winter time, but we had planned for 3 interesting speakers. Unfortunately, both Olawale and Joseph are calling in sick. So we have one of our great grantees with us today, Adéla, and she will be sharing her work. But before she does, I would like to say a few words on PAR, just to make sure that you are up to date with the work we do. PAR Foundation is working to help eradicate the spread of antibiotic resistance. And we've done so since the start of our work, 2017. We do have an audience and a global community of 1,000 plus passionate researchers and students working towards this goal. And with the funds that we are gathering, which is about 16 million Swedish crowns, we have distributed that into 25 projects since the start. Global projects in around the world, and this year we funded the first project in Latin America as well. So we are working on a global capacity to fight antibiotic resistance together with you. But let's dive into today's agenda then, and we'll have the pleasure to welcome you, Adéla. I would like to hand over to you to do a short presentation of yourself. You work in Holland, in Groningen.
Yes, hello everyone. My name is Adéla. I work now in the Netherlands, in the north of the Netherlands, but I'm originally from the Czech Republic. So I changed countries within the Europe. And I'm a researcher in biophysics and chemical physics. So I use methods of physics to understand how bacteria and antibiotics work.
Super. I will hand over to you. I'm excited to hear about your project. So I will stop sharing and then you can take over.
Yes.
And then let us hear from you on your work that you do.
Good. I will tell you today about the project that I did before in the last 4 years, basically. So it's a 4 years progress we made, and I will take you on a journey how we discovered that organization of bacterial outer membrane can be a viable target for antimicrobials. On the background image is resistant bacteria Staphylococcus aureus. And I just changed the pointer. So we have the gram-positive resistant bacteria Staphylococcus aureus, which is now considered as untreatable and deadly. It infects people with open wounds after surgeries and accidents in hospitals worldwide. And you all know, because you are here on this seminar, that antimicrobial resistance is a huge problem worldwide. Actually, in 2022, there was a first study that showed the amount of the impact of the resistant bacteria on human deaths worldwide. It was published in 2022 in the Lancet, and according to this study, that they showed that in 2019, in a single year, antimicrobial resistant bacteria infections actually played a role in almost 5 million deaths worldwide, and these numbers are increasing. So we have to fight this problem, and my approach to fight it is to use the methods of physics and chemistry to really understand how antibiotics and their targets, how they interact in on a molecular level. And today I will talk about this small molecule, which is called AMC 109. AMC 109. It was fabricated in a pharmaceutical company Amicode in Norway, according to a minimal pharmacological model of a working surface active molecule, of a working antimicrobial peptide. According to this model, such a molecule has to have some portion which is hydrophobic, which means that it repels water. It does not want to be in water. And some portion needs to be hydrophilic, which on contrary likes to interact with the water surrounding. This antibiotic is active against broad range of bacteria. It's successful in clinical trials. It does not show resistance generation so far. So far so good, it works. But when I contacted the pharma company and I asked them, so what it actually does to the bacteria, how does it kill them? They told me, yeah, you know, it somehow disrupts the membrane, but we do not know really how. So my question is, how does it actually work? And if we understand the working mechanism, can we maybe move forward and design some applications of it? I work mainly with bacteria Staphylococcus aureus. Here is a schematics of the bacteria. At the outer side, it has a cell wall, and important part of the cell wall here in the inner part is a membrane. And the membrane is the target of my antibiotic. The membrane consists of a double layer of lipids, and it also has some compartments which are slightly thicker than the rest, and these are called membrane domains, and they are very important in living bacteria. The domains are important for signaling, communication of bacteria with the outer surrounding. The membrane is important for maintaining the stability of the bacteria. The domains are the place where the bacteria synthesizes the material for the whole cell wall around, and they are also the place of resistance to methicillin and penicillin. And the membrane as a whole is the target for my antibiotic. So my research question now was quite simple, more focused. How does AMC 109 affect the membrane of bacteria? And to answer this question, I employ a variety of biophysical methods that complement each other. First, I use classical atomic force microscopy to image the membranes of bacteria. Here on this microscopy image, the black part is just the hard surface. And on this hard table, basically on a hard surface, we have membranes. These yellow-orange patches are membranes which are extracted from bacteria. So just the pure membrane, not the full bacteria, just their outer membranes. And I put them on the hard surface and they lay flat. So I can image their surface and how it changes when I add the antibiotics. I can see the dynamics of the changes in time with high-speed version of atomic force microscopy. And to see really the molecular level and atomistic interactions, I employ computer simulations to really see how the antibiotics interact with the membrane. I would like to mention that these experimental methods have a huge advantage, which is the resolution. We can go to sub-nanometer levels. Nanometer is 10 to the power of -9 meters. So we can really go to the sizes of individual molecules, and we can see individual molecules, how they move in the membrane and which molecule to interact with what. So these are my experimental approaches, and let's go to the results. Our first question was quite simple. What happens with the antibiotic molecule if we add it into water? So we have water solution. What happens with our antibiotics? And if we remember the chemistry of it, it had some part which was hydrophobic, repelling water, and another part, hydrophilic, likes to interact with water. It's not surprising that these interactions drive formation of stable aggregates. We have these micelle-like aggregates where the hydrophobic parts, which are here, this light blue, they are hidden inside. So the contact with water is minimized. And only the positively charged parts are sticking to the exterior and maintaining the contact with water. We put these molecules in our computer simulations to the membrane. Here the membrane is this bilayer of lipids, and you see that we have two different kinds of lipids in our model. There are negatively charged lipids, which are red. Negatively charged lipids are typical for bacterial membranes, and there are neutral lipids, which do not have any electric charge, and these neutral lipids are common in both human cells and bacteria. So if we have this membrane with some portion of this negatively charged lipids, we are mimicking the membrane of bacteria. And here you will see a short video from the computer simulations, how the antibiotic attaches to the membrane. You can see in this video that the micelle first comes. It sits on the place where the red negatively charged lipids are, so these lipids that are only in bacterial membranes. And then gradually it remodels the antibiotic micelle aggregate, and it dissolves all the antibiotic material inside the membrane. Important thing here is that it does not interact with neutral membranes. So it does not interact with the human cell membranes. So the fact that it aggregates and sticks these positively charged molecules outside, positively charged residues outside, it drives the selectivity for bacteria over human cells. So now we know that our antibiotic attaches only to the bacteria cells. So let's have a look what it does to their membranes. And for that, I employed the atomic force microscopy. Here is a depiction of it. It's like a tiny needle that we scan the surface with. We like touch it like with a finger, and we can get to this really molecular size resolutions. Here on this image is our membranes extracted from bacteria, flat on the surface, and we add the antibiotic to it. You see that the membrane patches here, they grow in size, and they also are thinner. If we add even more antibiotics here, the membrane grows even more, stretches, spreads over the whole available surface. I did the test study, the very same experiment with the well-known pore-forming peptide from honeybee venom, melitine. This peptide is also antimicrobial peptide, and it's known to also affect membranes and make holes in them. And that's how it actually kills bacteria. It just like makes it like an emmental, makes holes in the outer structure. If I perform the very same experiment with AFM here, this is what happens to the membranes of bacteria. You see it's full of holes. But this does not happen with our AMC 109 antibiotic. So we can clearly conclude that AMC 109 does not make pores or perforations. It somehow attaches inside, goes inside the membranes, stretches them, but does not make any holes. And this was kind of intriguing for us, because if you read literature and like go talk to researchers, then like if you have membrane active antimicrobials, usually they say, yeah, it disrupts the membrane, it tears parts of the membrane out, makes holes in it, and this is how it kills bacteria. But our antibiotic here does not do it. So I was searching like what it does to the bacteria that it kills it, because the membrane is still there, it's still intact, right? Only like stretched. There is more material, it's stretched, but it's intact. And we believe that we found the answer to this question, like what it really does to the membrane, again in atomic force microscopy, but in the high-speed version of it. If we look at the membrane before adding any antibiotics, we notice that there are these bumps in it. This is a 3D model of this of similar image like this. You see here these bright dots. These are portions of the membrane that are higher in a thickness. This is a 3D model of the membrane. These higher parts of the membrane, these are the lateral domains needed for the signaling, communication with outside world, building of the cell wall, and resistance to methicillin and penicillin. And I noticed that if we add the antibiotic to it, the membrane stretches and the domains are not there anymore. So we had a look at this phenomenon closer in time using the high-speed AFM, and this is the video. I try to run it. Yes. So you see these bright spots in the membrane patch. They are moving very fast. These are the domains moving around. And we add the antibiotic moments ago. And after a while, you see that these domains, they slow down their movement and start clustering together in the center and at the edges. And after another minute or two, the domains start disappearing. The membrane spreads and the domains dissolve into the surrounding membrane. So they are gone and the bacteria cannot use them anymore. And without the domains, the membrane is not functional. Sure, there is still the barrier function that no inside liquids can leak out of the bacteria, but all the other functions of the domains are gone. I was searching for some other molecules, other antimicrobials, anything what would resemble this new mechanism that we found. And actually, we found out that disinfectants, small disinfectant molecules, they actually work very similarly. Here we have benzalkonium chloride. It's a disinfectant that is used on a daily basis in many hospitals just to disinfect tables and equipment in hospitals. And if I have a look what happens to the bacteria membrane, here I have the bacteria membrane, I add the benzalkonium chloride, this disinfectant, the domains cluster together now, and they slowly dissolve into the surrounding membrane and the membrane spreads. The very same thing as without antibiotic AMC 109. If we do the very same experiment with ethanol, very known disinfectant, before the addition of ethanol, we have smaller membranes with domains. After the membrane is stretched, the domains are gone. So AMC 109 acts as a disinfectant. And disinfectants are very powerful molecules. They are used for centuries already. Ethanol, it has been for, 400 years without generating any resistance. The problem with disinfectants is you cannot use them as antibiotics because disinfectants kill everything. Ethanol also harms human cells, so you cannot use it as an antibiotic. But with this antibiotic in hand, the fact that it aggregates in water and is selective only for bacteria over human cells, we actually have a disinfectant which is highly selective for bacteria. And this story has been a 4 years research work of 3-4 different research groups with me managing the project, and it was published earlier this year. And I'm now continuing this research at the University Medical Center in Groningen with the support of the Power Foundation research grant. My idea is that we dissolve these lateral domains here, and they are the place of the resistance for methicillin and penicillin. So if we dissolve them, we will possibly reverse the resistance to penicillins. And this is ongoing research. I'm just now doing the very first experiment on it. So hopefully there will be more exciting results coming soon. And with that, I would like to thank all the people involved, my home group at the University of Groningen, the people who did computer simulations, and people responsible for the extracts from the bacteria and their analysis, also for funding that research council, and pharma company Amicode for providing the antibiotic for my study. And thank you for your attention. If you would have any questions, I'm happy to answer.
Thank you, Adéla. I think this is a very exciting project. And what if you were able to reverse a resistance to that penicillin? It's very, very tempting and lovely idea. Gabriel, do you have a question there?
Yes, so there's a question. How are the membranes extracted from the bacteria?
So
But
how were the membranes extracted? So there are really common procedures already. To do these things, I have to dig into literature that is 100 years old. And it's a microbiology and biochemistry essay that you just grow bacteria. We have to be very careful because we are growing resistant bacteria. So we grow them in this biosafety cabinets. And then after they are fully grown, we kill them. I boil them in a soapy water, basically, kill them, and then we extract them. We extract portions that are dissolvable in water and that are soluble in methanol. And so we add different solvents and we separate different portions of the bacterial material. So we, in the end up only with lipids extracts, and lipids are molecules that are kind of at the edge between water and oil phase. So we can extract only the lipids. Then I store them in a freezer, only the lipids from bacteria. And when you put these membrane lipids into water, they form the membrane spontaneously because it's their like a low energy configuration.
okay, yeah, that's a question. Thank you.
I see that there is another question as well.
Actually, I see the questions. So here it's if the bacteria are effective against gram-positive or gram-negative. Actually, they did test it in the pharma company Amicode. It's effective against both gram-negative and gram-positive. I think the antibiotic is so small that it just goes through the cell wall also of gram-negatives. They have like more barriers around, but this antibiotic can slip through. And so it is effective against both. And what is important, it was also tested against, I think, like 70 clinical bacteria strains of resistant staphylococci and streptococci, and it all was working well. So it's effective against these resistant strains too.
Super. And if you have questions, please post them in the chat. It's the easiest way to get the questions answered. I saw someone raise their hand, but if you type your question in the chat, Adéla will be able to answer. In the meantime, then I will actually just ask you, do you see others working on the same or similar work that you've done, or is this quite unique, you'd say?
I would say it's quite unique because many microbiologists work on antimicrobial resistance and like looking at the mechanisms of antibiotics. but I'm a physicist, so I look really at the molecules and atoms, how they interact. And this is not really common, I would say. More they look into like how the antibiotics changes the pathways and the metabolic response of the bacteria, which is another approach, I would say. And this is one thing. And second, for the majority of resistant infections, actually gram-negative bacteria are responsible. So this coccus bacteria are more neglected because there are not so many in this like list of resistant bacteria strains. So not many researchers actually work on these gram-positive ones, on the cocci. So I actually found this quite interesting too, that it's kind of a niche that not many people focus on it, but it's also important because these staphylococcus infections, and not only that, but also Streptococcus pneumoniae, they can be deadly, not treatable. So it's also important to study those, although they are not so abundant, I would say. Yeah.
No, you're right. And I also think it's interesting to work across disciplines because you have a different view and another eye towards the problem. So I've seen that in other projects where researchers work across disciplines and collaborate. And I think that's when you get new or unique or different results.
I work with chemists, biochemists, microbiologists, and also just started at the hospital environment working with clinicians. So I have also insight from them, which is very interesting to get all the other point of view. Yeah.
Nope, I agree. Well, thank you so much for sharing your insights with us today. And I really look forward to follow the work that you do. We'll hope to get back to you in the spring or summer, I would say.
Yes.
Super. Well, as I was explaining to everyone before, we had 2 of our participants got sick today. So Adéla's presentation will actually be the one that we do. We hope to get back to Joseph and Okere. So I will thank all of you joining us today and hope to see you with us in the spring. This will be the final seminar and webinar that we do this fall. We'll take a winter break and we will be back in the spring 2024. So a big thank you for joining us. If you have ideas of what you would like to hear, please let us know. And otherwise, I thank you again, Adéla, and wish everyone a great continuation of the day. Thank you so much.